It's a great pleasure to have Professor Stéphaine Baud here from University of Reims. I apologize for my pronunciation of the name of university. Professor Stéphaine worked with simulations of biological systems, especially protein systems. And hopefully you are going to have the opportunity to review some of the concepts you have been discussing in our course of machine learning. Professor Stéphaine, thank you very much for accepting participating. It's an honor to have you here. Thank you.
Thank you very much, Walter. So, good afternoon to everyone. It's a real pleasure to be able to present some of the work we've been doing in the past year in the team. So, as you were told, I work in a research unit which is called MEDIC for matrice extracellular and dynamic cellular. So, extracellular matrix and dynamic extracellular matrix dynamics. So I will try to tell you about this very complex object and try to show how with molecular modeling and in some cases with deep learning or artificial intelligence, we try to understand how these elements behave in a very complex environment. So just a few words about the biological context. The focus of our research unit, which is composed mainly of experimentalists, and we have a small team focusing on modeling and also on imaging and all the analysis we can perform on that. So the topic of interest of the unit is the extracellular matrix. So the extracellular matrix is a very complex middle, which is the interstitial middle between the cells. And there are a lot of different objects that we can find in this medium. You can see here pictured that you can have glycoprotein, carbohydrates. You can have very long fibers in different forms. We have glycolipids. We have also the cell membranes to take into account. So this accounts for very different objects. So the extracellular matrix is characterized by its diversity, the fact that it's composed of structural protein that can form fibers. You have protein polysaccharide complex. You have what is called adhesive glycoprotein. So all these elements, they can maintain the tissues, and they also are here for the cohesion of the tissues. This is extracellular matrix is a reservoir of growth factors. It's also implicated in many cellular processes, in particular cellular regulation.
Stéphaine,
And yes?
Sorry to interrupt you. I guess you are not sharing the full screen.
Oh, sorry. Okay, you're still on the first page. Okay.
You will see on the first page.
Okay, I will share the whole screen. Sorry for that.
It happens to me all the time.
Okay, so here is this the good one? It's because I guess I have presentation.
Now you have it. Thank you very much. You can go.
Yes. Okay. So you have the one with so just here. This is the second. Okay, the third page. Did you see this one?
Yeah. Yes.
So I'm on slide 2. This one. Did you see it before?
No, it's first time.
No. So okay. So just I won't say everything again, but here you have the representation of the extracellular matrix, and you can see here that you have a lot of different objects that are represented. So this goes from here fibers, which is in green. You have like glycosylation. You can have carbohydrates on the membranes and so on. So diversity of different proteins. Just to be sure, you can see slide 3. Right?
You can see it.
Okay. So again, we have a lot of diversity in these extracellular matrix with different roles. And so here is a small representation of what we saw just before. And in terms of scheme, another way of representing the challenges we want to assess with molecular modeling, here you have another picture or another representation of what are the main elements we've been studying in the extracellular matrix. So because this is the limit between the intracellular space and the extracellular space, of course, you will have the membrane. We can have here transmembrane protein. And this transmembrane protein, they can bear modifications such as glycosylation, but they can also be degraded, these one other proteins. And so they can interact with other small part of protein, which are here some degradation products, what we call in the lab matrikins. So matrikin is something which is very specifically, that was specifically defined in Reims something like 20 or 30 years back. And these are the products of degradation of different extracellular matrix protein. So what we try to do, what we do in the team is we have some people focusing on molecular modeling because we strongly believe that this is a dedicated tool for performing structural bioinformatics. And so what we try to do with our colleague from biology and biochemistry is to bring something that we call a computational microscope, which gives us access to atomic and molecular vision of the different molecules we are interested in. So I'm not saying these are just the only axes that we are developing, but mainly in relation with this molecular modeling field, we have three main axes of action. The first one is the one which consists in studying the structural characterization and the structural activity relationship between peptides and other proteins. So the methods that we use are molecular dynamics, for instance, energy minimization, normal mode analysis. Second axis we work with is an axis that aims at characterizing the interaction between matrikins and extracellular matrix protein. So we can perform homology modeling. Of course, again, we can perform molecular dynamics, but we also perform a lot of molecular docking experiments. And the third axis is one that we try to really dedicate some amount of time, is an axis related to methodological developments. So we try to bring a new method to optimize computational distributions, for instance, new method of analysis and representation. And so these different axes, of course, what is necessary to perform good research with them is to be able to discuss with experimentalists, and so to understand what they need or what they want to understand from the different system. Of course, we have to perform extensive analysis, and also it's important to discuss with the experimentalist, because if we want to choose the proper methods and models, we have to understand really what question they want to answer and what they already know about the systems. So, as I told previously, not all the results I will present are related with artificial intelligence, but I will try to bring some elements or present previous results and see how we came to use at some point deep learning and artificial intelligence. So the first example I would like to present is something which is related to the design of anti-tumor matrikins. So within the different elements of, or within the different proteins of interest that characterize the extracellular matrix, There's a protein, which is a fiber protein, which is collagen. So collagen, you have a lot of different types that you can find in organism. One of the collagen that can be found is collagen of type 4. So the 3D structure of collagen is really specific. You will have a part that will organize in a triple helical domain, and you have an N-terminal domain, which is not necessarily structure, and a C-terminal domain that is said non-collagenic because it's not structured in a triple helix, and in general, it's not very well organized. And we have a team in the lab that is working on all these, that is interested in how collagen can mediate, let's say, tumor progression. And so previously they could show that a chunk of collagen that is named tetrastatin was able to display anti-tumor properties. And so if you can see here, you have tetrastatin, which is a very long part of collagen. Of course, collagen is very long, but here tetrastatin, you have 230 amino acids. And so what they started to do experimentally was to cut kind of randomly these tetrastatin to see where they would stop to get some anti-tumor effect. So here the color display the property with respect to the anti-tumor character of the peptide. So when it's red, it's not working. When it's green, it would show anti-tumor properties. And at some point, we've been discussing together, and instead of cutting randomly, we had a look at the sequence and also performed some different experiments on molecular dynamics and also docking. And at some point, running in parallel experiment, wet lab experiment, and also in silico experiment, we could come to show that a short peptide, 30 amino acid peptide, with this sequence here, would have interaction with integrin, alpha V beta 3 integrin. And so well, that was interesting because alpha V beta 3 integrin is a protein that surexpressed in melanoma. So we've been investigating both experimentally and also in silico what would be the key features of this interaction between the integrin and this what was called QS13 peptide. So what we've been doing from the in silico point of view, first we would need to decipher a bit the structure and also the flexibility of the peptide, because even if it's a short peptide, the fact that you have very large side chain of this peptide and also you have 13 residue makes it difficult to study it properly with a full number of degrees of freedom with regular docking experiments. So the fact that we could be able to sample the conformation explored by the peptide would give us insight then for the docking experiment on how we could, let's say, restrain the backbone to perform better experiment. So here, just quickly, I will explain quickly. We ran simulation, MD simulation on the peptides. So of course, we consider the classical peptide, QS13. When we look at the sequence of this peptide, we see that there's 2 cysteine residue. And this cysteine residue, they can form disulfide bond. And the experimentalists could show that, in fact, this bond could be formed kind of instantly. There was no... It was very easy to get this formation. So that's why we perform simulation with and without the disulfide bridge to understand how it would change the conformation. And also we run some experiments with mutation along the sequence to try to understand how the side chain would modify or could modify the conformation. So we did like 200 nanosecond of molecular dynamics simulation in explicit water model. And then we could compare the conformation that would be obtained. Here, what I show are superimposition of the trajectory of different snapshots of the trajectory of the classical QS13 peptide with the disulfide bridge. And what can be seen here is that this disulfide bridge, of course, restrain this part of the peptide, but then the effect here is that it exposes the arginine, which stays accessible. And of course, if we superimpose now the main conformation obtained through different simulation here, all these simulations are with disulfide bridge, but you have some mutation at some point. You can see that the mutation at the beginning of the sequence or just before the cysteine residues, around the cysteine residue do not impact, but here, of course, when you change the arginine in the lananine, you lose this occupation here. So it was already something nice to understand how this disulfide bridge would constrain the conformation. And then from this simulation, we would be able to perform what we call clustering of the trajectory, which means we would be able to extract the main conformation. And that would be of great use because then when we want to go through the characterization of the interaction between QS13 and integrin alpha V beta 3, with Autodoc software, we use a lot this software in our lab. We have to take into account the fact that we cannot go above 32 degrees of freedom, so we cannot consider more than 32 torsions within the peptide when we perform docking experiment. So we used the molecular dynamics simulation performed before and the clustering performed also on this molecular dynamics simulation in order to perform what we called semi-flexible docking, which means that we would freeze the backbone and some other specific groups such as guanidinium when needed, and we would let the side chain free of exploring the different conformations that were accessible at the surface of the protein. And so when we perform this docking, since integrin is a very big protein, instead of doing one blind docking over the whole protein, what we did is we cut the protein in 180 different overlapping boxes with a spacing that would allow very good, not very good, but a good level of energy resolution. So we accumulated a lot of solutions with these experiments. We could accumulate 27,000 solutions, and then we restrain our analysis to 180 best lowest energy. What we did is we looked at the distribution of this solution on the surface of the integrin, and we could identify 5 preferred areas of interaction. Okay, and so here you have the percentage of solution that can be found in each of these areas, and also the best free energy of binding that is associated with this area. And what was interesting with this is that one of the area is common with the interaction of the RGD peptide that is found in the crystallographic structure. So what we did is in this area, we did focus on the interaction that would be made between each subunit of the integrin, either the alpha V or the beta 3 subunits. And we could evidence some contacts that are common with the one that make the RGD contacts, the RGD peptide with the integrin. So what we could, let's say, keep in mind from this analysis, of course, is that docking was able to highlight different area of interaction, but also could highlight that the presence of the disulfide bridge is very crucial for the interaction with the integrins, and that some of the residues are common with the interaction made with RGD. And so, at last, to validate what we obtain as results with the in silico experiment, our colleague, the experimentalist biologist, they perform experiments, so in vitro experiments that I don't show here, but they could perform experiments showing that the QS13 peptide was compete with RGD peptide, showing that one of the region we evidence is validated experimentally. And the other set of experiments that they run is that on mice, they could study the effect of injection of either some control solution or solution containing tetrastatin or solution containing the peptide. They could control over time how the tumor volume would evolve. And what you can see here is that clearly already tetrastatin shows some anti-tumor effect, decreasing the size decreasing the volume of the tumor, but QS13 has even a stronger effect. So with this work, this common work with the experimentalists, we could show that QS13 has an important role First, the presence of the disulfide bridge in QS13 is very important, since it brings constraint that will be very important for the interaction. Then we could identify 5 areas of interaction. And of course, we could also identify key residues implicated in the interaction. And so this peptide has been validated through different publications. And there's a PhD student now that is working both on the in silico level and experimentalist level to try to improve the solubility of this peptide. And so we try to bring some mutation in this peptide to keep its interaction with integrin, but to favor a better solubility in water so that it could be injected more properly. So you could see here an application of what we called regular docking or classical docking. Some years ago, because we are working on extracellular matrix, one question was raised at some point by our collaborators. You can see here, we could show that QS13 interacts with integrin. It was shown in silico, it was shown experimentally. But the thing is, once you have some molecules, such as the small peptide that are circulating in the bloodstream or in the body, what assurance can we have that these peptides do not interact with other proteins? And so that's how the idea of molecular inverse docking came. So what is inverse molecular docking? So as shown previously, we can perform classical docking in general. That's what's done in most in silico experiments. You know some protein and you want to test different small molecules or ligand on this protein. And so you do some blind docking or some targeted docking on specific areas. And with these matrikins, we had this question of, is it possible to see if ligand of interest is able to interact not only with the target or the known target, but also with other proteins of the extracellular matrix or of the organism. And so the idea of this inverse docking protocol is to be able to test for one small molecule how it can interact with the database of various proteins. In addition to this question, we wanted to take advantage of, let's say, a supercomputer in a way that we could be able to use the number of CPUs, or a huge number of CPUs that are that we have access to when we can use a supercomputer. So, in addition to be able to perform experiments in parallel for different proteins, we thought also that we could have access to, that the fact that we would have access to different CPUs could be used as a way of cutting the proteins in different parts, so with different boxes, with overlaps, so that we don't forget any region. And once we have defined these different regions, so we have what we call some arbitrary cutting. So we cut the space regularly, or we could use some server to search for pockets so that we can target on some specific area. But once we have defined these different regions, then we have access to supercomputers, and we can distribute the different docking experiment on various CPUs. So that's how we came to develop a few years back what we called automatic molecular inverse docking engine, that was also named AMID. And so this AMID protocol could be validated on databases of globular proteins. So we were able to show that for a given ligand, using some arbitrary cutting, what we call here grid or the search pocket, or that if we associate the two of them, we would be able to screen and identify a selected number of proteins that would be interesting for the ligand to interact. Also, we were able to show that this method of docking would outperform the classical blind docking experiment and would allow to search for solution, let's say, faster and more efficiently. So this method was developed with CPUs, and so was used with, this protocol was developed with the compiled version of Autodoct on CPU. And a few years back, with the COVID pandemic, we wanted to use AMID architecture to speed up the search between databases of ligands and databases of protein. And so, discussing with collaborators, we came across the compiled version of Autodoc, the GPU compiled version of Autodoc. So we transfer the protocol to GPU architecture, and that's how came this AMID v2 version. And with this version, what we were able to do is to accelerate the process of the search. As you can see here, we really outperforms with the v2 version the results of the v1 versions. It's one GPU in terms of execution is almost similar to something that was performed before on 300 CPUs. So this is for one ligand versus 87 proteins, and with 9 ligands on the same database, we can see here that we really decrease the time. So if we have access to one GPU, of course, we have a certain amount of time, but still it's in minutes, and we drastically decrease this number of, this time of execution when we increase the number of GPU. Of course, we checked that we didn't lose anything in the precision, in the performance of the docking in terms of position of the ligands. You can see here the pose obtained with the old version of AMID and the new one. So they are rather similar. And so, again, we could validate this methodology. Just a quick here overview of what we gain in terms of time using this GPU architecture. So you can see here that on a desktop computer with 4 CPU, we could treat 20 ligands per week. So if we have access to 448 cores with the old version, we could treat 100 ligands per week. But then when we go through GPU, we can really increase drastically the number of systems we can treat per week. So that's what is really interesting for us, and that brings us to the notion of high throughput screening, not only one ligand versus a database of protein, but here we can confront now a database of ligands, huge one, and a database of protein. And so now with this notion of docking, you can see here that we went through, we started from one docking experiment on a whole protein with just one ligand. We moved with the CPU and then the GPU to the possibility of exploring confronting a database of ligand with a database of protein. And now there's the question of going further, or being more efficient in what we do. And that's how we come to the notion of the prediction of protein ligand affinity with heterogeneous graph convolution neural networks. So this part that I will show you is the results of the work of a PhD student who defended his, so he's now a doctor. He defended his work at the end of the last year. And I will show you what he did in relation with the scoring function and with the development of a new scoring function using deep learning. So just a few words about the graph and convolution. So I'm not an informatician myself, so I understand the background that is behind, but really this is, Kevin did the work of implementing this notion in the tool he developed. So a graph is basically an ensemble of nodes, and these nodes, so in our case, that's what I will show in a few slides, the nodes will be atoms, and these nodes, they will be linked by what we call edges. And so in informatics, each node and each edge can contain information, and it is said that they may be augmented by features. So this is how we define the graph, and then the notion of convolution. So graph convolution is something that extends classical convolution to graphs. So a set of nodes linked by edge, and there's a notion of information that is shared through the edge. You can see here for this white dot, the information that is bared by this dot will be let's say, implemented or will take into account, updated with the information that comes from the neighboring edges. You have the same representation here, or another way of representing this is the convolution, which consists in defining the information bared by the nodes, and then from these star nodes, you will update the information it bears, taking into account its neighbor. And so you can repeat this like a certain number of times and taking into account different levels. So you will bring this as far as you want to make converge the information. So Kevin, for developing his new scoring function, treated what is called a heterogeneous graph, meaning that you would have one graph here in blue, a second object that would be defined by a second graph in green, and then there would be the possibility to have some kind of crosstalk between the graph. So you create new edges that connect the two different graphs, and here you have the violet arrows. And then you have to mix the information that are brought by each graph. So what you do is you will have information from node of graph 1 to nodes of graph 1 that will go through these specific edges. Then same thing within the second graph, and then you will have some information that will pass from graph 1 to graph 2, or reversely from graph 2 to graph 1. So you have to collect information on nodes of graph 1, graph 2, and then this information from graph 1, they can go on through graph convolution layer on graph 1, but also on graph 2. And then, of course, graph 2, you have this for itself, but you can have the reverse. And then you have at some point to perform some kind of aggregation on the nodes. You can activate, and then you repeat the process a certain number of times. And so what does it mean from the molecular point of view? Here, to define or to elaborate the new scoring function, we had to collect data, and these data were collected from molecular complex, okay? So we have to transform this molecular complex to heterogeneous graph. So how did we do? We have here a protein that interacts with the ligand, and so we define a region around the ligand at 10 angstrom of any atom of the ligand, that's the red part, as the interacting graph. And so now we can define two graphs, the one from the protein. So it consists, the nodes are the atoms of the protein. The edges, they are the bound, okay? And then we have a graph that represents the ligands. The same, the atom of the ligands, they are the nodes. And then the edges are the links between the atoms. And then we can define the heterogeneous part, which links atoms of the protein to atoms of the ligand. What about the features, the information that the nodes can, or the edge can bring? So here, there are mainly physico-chemical features that will characterize the type of atom, the hybridization, the charge, the fact that it's hydrophobic or can be aromatic. It can give it can be H-bond acceptor, donor. And for the links, the bonds or the edge features, you have something that which characterize the distance, the fact that it can belong to an aromatic cycle or to a ring, the type of bond, single, double, or triple bonds. Is it something that links hydrogen atoms? Do you have something that is characteristic of a salt bridge, and so on? So you can see here that these features, they can be either Boolean or they can be real numbers. It will depend on what is characterized. So this is information that is put on nodes and on edge. And so with all this information, the methodologies that Kevin developed, so edge score is based on heterogeneous graph convolution. And so we have to perform a convolution on the atomic part and then on the molecular part. And then we have to perform a final prediction that will give us something that is related to experiment. So here, for the atomic part, it's just something specific now with the protein and the ligand, but it's mainly what we saw previously on the heterogeneous graph convolution. So we have some information on the nodes, protein or ligand. Then we can perform convolution, okay? Then we can aggregate, activate, and then we can start again. And so we do that for a certain amount of cycle. Then at some point when we are over, or we finish the number of cycles we wanted, we have something, an information that is embedded within the atomic part of the protein and the ligand. And so we can start from this embedding on the protein and on the ligand. Then there's some informatic part that is called pooling. There's another graph convolution that is performed, another activation, and then we'll restart this element a certain amount of layers, a certain amount of time. And at some point, we obtain some molecular embedding on the protein and on the ligand. And the final part, which consists in the prediction, we take this information on the protein and on the ligand, we mix them, we get what we call an interaction fingerprint, some molecular layer processing, and then we obtain, through mathematical function, a score that is trained to represent the scoring function that is given by the experiment. So what we did with this new scoring function is that it was trained on a set of structure extracted from the PDB bind, so almost 20,000 structure. The score that was calculated and compared with was linked to the experimental affinity that we also call the ground truth. And so we did that on a specific material on the supercomputer we have at the university. So just for your information here, this training on this specific setup to train the model, it takes almost something like 3 hours. So kind of efficient, let's say. And then once we could have a scoring function and the method was trained, we tested the method on another data set, which is the CAT 2016, that is composed of 285 complexes. And so we computed different scores of the crystallographic poses, and then we could associate to that different metrics, coefficient of correlation, standard deviation, mean absolute error, root mean square error. These are different description of the differences, let's say, or the differences between the ground truth and so the scoring, the ground truth pose and the recalculated pose. And so the method was compared both to classic and other intelligent artificial methods. So here you have the results obtained with classical methods, with other artificial intelligence method, our methods. And here is the aim of each of the parameters that was calculated. So RP, we aim for value close to 1. SD, we want to go as low as possible. The same for these two other metrics, okay? And when we can see here is that with our method, we outperform all the classical methods, and we are among the best method of the artificial intelligence methods. We could also look at some kind of linear regression between the score we predicted and the targeted score, which means the experimental score, the one obtained from the experiment. And also what we were able to do for this set of experiments in general, there are some decoys that are proposed. And so we looked if each score was able to detect the ground truth from these decoy poses that were proposed. And so here what we can see is that each score find the ground truth as the best solution among the decoys 30% of the time. The ground truth is in the two first solution among the decoys half of the times, and the top three or the three first solutions 60% of the time, which is kind of good compared to other methodologies. And so here you can have some illustration and comparison of the results. Sorry, I just realized I didn't put the PDB ID here. So here you have the superimposition of the three first poses as ranked with our methodology. And you can see here that among the decoys, each score ranked the ground truth as the second one. So this is a short peptide. We see that there's not that many discrepancy between the position. When the size of the, sorry, not the peptide, the ligand, when the size of the ligand is increasing, here you can see on this example that the ground truth is not ranked within the first solution. But here the scores that are given among the decoys, they are very, there's some difference, not striking, but good difference between this score and the one of the ground truth. But here we can see there's some overlap, but there's some kind of shift, or let's say it's not in the same direction for this ligand. So we might have to investigate this effect at some point. So here, with this new methodology, what we could show or validate is the fact that AG score is what is among the best artificial intelligence scoring function. And here, what we hope is that by splitting the learning according to age class, this score will be a good candidate for transfer learning, meaning that we could adapt this to other type of docking experiments, meaning now with extracellular matrix, we really would like to go towards docking of glycosylation on proteins or glycosaminoglycan on proteins. And since here we work with class of ages, we think it's this methodology is a good candidate. And also here, what now is missing and we would like to do is to integrate this score in a complete workflow of protein ligand molecular docking. Just to finish, because I'm over time here, just to show you some other projects we have in the team. Here I already focused on the molecular aspect of what we do and the application of deep learning with this new scoring function. In the near future, we would like to use also artificial intelligence in process that we called mesoscopic description scale. When we study extracellular matrix, we have a lot of different molecules to take into consideration that will interact. And if we do that with atomic scale, it will be very difficult, even with very powerful computers. So one tool that was developed in our team in the last few years was this mesoscopic descriptor. Based on biochemical information or biological information, most of the protein of the extracellular matrix can be described as being composed of different modules. And so for each of these modules, most of the time we are able to find some information in the PDB or either through X-ray experiment or either cryo-EM or cryomicroscopy experiment. And so the idea then is to rebuild the molecule, assembling these different blocks. And so that's how came the idea of using rigid body dynamic, saying that assembling this block, we can reconstruct big proteins such as collagen or laminin or even glycosaminoglycan such as pelican. And you can see here, these are the reconstruction we get starting from experimental data. And here is the representation that you can find in the textbooks, regular textbooks. So you can see here that in general, people, they don't have any information with this representation of the real scale of the objects. We have here the real scale of the object, and you can see that these objects span on nanometers scale. And again, it's very difficult to assess the interaction of this element, or it would be very difficult to assess this with regular modeling. And so once we have these blocks, here we have this description. So for each of the blocks, we will have some rigid body that are linked through links, and then we just have to take into account the environments with Langevin dynamics. So we integrate viscosity and random forces to have the effect of the cellular environment around. For the protein that are anchored to the membrane, we updated the version we have with the possibility to the integrins here to move perpendicularly in the membrane with this impeller force field. And so then we can put everything in a box and see how this ensemble moves and interacts. So here you have the oldest version of this mesoscopic engine So you have the integrins here that are attached to the plane, the white plane, which is the membrane. So the membrane for now is rigid, but we've started to have something that is more flexible. And so you have here collagen, you have perlecan in cyan, you have nitrogen and laminin. And so you can see here, we have the possibility to let everything move and interact. But with this, we also have the possibility to interact and kind of grab some of the molecule and see how they react to being extracted or to being pulled in other molecules. So for now, this is just with the rigid body mechanics, but at some point, if we can get some information for experiments, extract information with artificial intelligence and inject this in this model, we really think we will be very powerful in assessing the main properties and see how these different elements assemble within the extracellular matrix. And so I hope you will excuse for being a bit late, but this is my last slides that conclude this presentation. So in our team, what we do is modeling, and we could see that it offers a wide spectrum of techniques, including artificial intelligence, and that with this, we are allowed to decipher and improve the understanding of biological behavior at different scales. But of course, the models will be reliable if we are able to integrate observables at each different scales. And so that's the idea here. We have these rigid bodies simulation, and we hope that in the coming years, we will be able to update the information we have here and have a better descriptor with integrating realistic description of the membranes and also the characterizing the interaction networks. That's the main thing we hope to bring with the docking experiment and with this new scoring function we developed. And also, we hope we will be able to bring realistic description of collagen and elastin, or either also, let's say modifications such as glycosylation. And also at some point, we would like to be able to characterize or link this with imaging process or observation that can be also made on different tissues. Here you can see arteries that are, these images are obtained on synchrotron. And so at some point, we hope we will be able to bring everything together and to be able to compare these, knowing that here all the analysis of these different images are also performed using artificial intelligence. So with this, I would like to thank you for your attention. I hope I was within the scope, and here you have a nice picture of the people I am very happy and lucky to work with every day. Thanks a lot for your attention.
Thank you very much, Stéphaine. We really enjoy your talk, and I think we're gonna have a good talk. During the talk, you started having ideas, and right now I have a lot of ideas.
Okay.
First of all,
and please,
go ahead.
Yeah, if I don't know, but if there are questions, I would be happy to answer.
Yeah, I'm going to open this session for questions. And okay. Some very fast comment. You are about to finish one volume focus on docking, current medicinal chemistry, that you contributed with a paper, and also are going to contribute to the editorial. And right now I have an idea to propose another volume focus on the application of artificial intelligence to develop a scoring function. That's something that can talk later, but for next year, okay? But right now I have so many different artificial intelligence scoring functions. You can put all together, most of them, in one specific volume dedicated to this topic. And it's going to be great, hopefully. And you have any comment?
No.
And I'm going to contact with you. Yeah,
of course, I would be happy to help.
And you are open to questions. If any of the students have questions, we are going to ask Stéphaine. Anybody? You are so quiet. Everybody is sleeping, okay? Okay, go ahead if you have any question.
Professor, eu vou fazer a pergunta, fazer um comentário em português.
He is going to say in Portuguese. I'm going to translate to you. Okay, Stéphaine?
Okay, no problem.
Primeiro, agradecer e dizer que foi ótima a apresentação dela. E uma coisa assim que eu queria fazer uma pergunta é que ela mostrou uma parte, comparando a metodologia convencional com inteligência artificial e os resultados. E eu queria ouvir alguma experiência prática, que virou um fármaco de verdade, que virou um produto de verdade. Se tem algum exemplo que ela poderia trazer para a gente.
Obrigado. I'm going to try to translate his question. He said he appreciates your talk, and he asked if you know of any successful story about application of artificial intelligence to generate a drug that is in the market now. If you know of any result related to this success.
I'm not so sure there are drugs in the market now, but I would have to check. But we discussed this a few weeks ago with colleagues. Apparently, there were some experiments that of generating ligand poses or even new molecules based on the artificial intelligence, and some of them would be promising, but I'm not so sure they even went through, let's say, clinical assay yet. But I don't know, like this, I don't know of any molecules, but I know there are promising results from the in silico part and even like wet lab part, but just didn't go on a trial yet, clinical trial yet. But I think it could be fast within the next 2 or 3 years. We should be able to see that, I guess. That's my feeling, at least.
Devo acompanhar, né, Alonso? Thank you. Any other question? Okay, so now I have a question.
Okay.
I'm really impressed. I know some of the work. I didn't know all that work. And one thing that was clear from your talk, that you have a close collaboration with experimentals, with people who work experimental. And that's the advantage. So how is this relationship with the modeling and the experimental? You get the feedback or you get the data and you generate a model. How is this relationship with the people who work on the experiment?
Yeah, so until like 2 or 3 years ago, it was more like people would come to us and say, okay, we have this effect, or we see that and we don't know yet how to interpret, and so or how to decipher, and so we would need some insightful or some insight information from modeling. The story of QS13 is kind of like this, but we've been like 2 or 3 years ago, we started to say, okay, we would like to go in the reverse situation, say, like, we would like to try something and then be able to propose or ask you to show that what we see in silico could be proved also experimentally. So, for instance, there's how we say that in English? There's brevet. Do you know the name brevet? So there's an interaction that is known in the lab between 2 big proteins, and the idea was to find the sequence that would be the best for the interaction between the two. So in order to design a peptide, and instead of cutting as they did for QS13 in collagen, they came to us before, and we designed the protocol together, saying that first we did some protein dockings to try to focus or find the main interesting region. And then seeing that, we chose peptide and we performed simulation with this peptide, and then we could focus or find the interaction zone. So we were able to define a region of, let's say, 15 to 20 amino acids. And so far, they've been doing several experiments with different cutting, and also we did some mutagenesis on the protein to validate the interaction, and so far it's working. So I cannot give the sequence because we have this, I don't remember the name, but it's, it's when you deposit the ID and you have to protect it. Yeah. But, and also now with this mesoscopic scale, that's what we would like to do. We would like to try different proportion or, let's say, vary the flexibility of collagen and see how it looks like when how does it change the way of the way the molecule will interact and associate. So it's really something we would like to do, and we've been working with or discussing with people in Paris that works on with the neuronal extracellular matrix, and they are interested between the interplay of glycosylation and the assembly of the neuron, the neuronal extracellular matrix. And so here, what we should be able to do in the future is, say, run experiment with the mesoscopic scale and compare with imaging they are able to do. And so we will find a way of calibrating our model so that we can explain what is going on. So I would say, the paradigm was experimentalists would come for us and ask what's happening. And now we try to work at least together and define the protocol together. And at some point, we would like to be able to come and say, okay, we would like to test this possibility.
Thank you, Stéphaine. Anybody has any question? I'm glad to translate if you don't want to make the question in English. You could also make the question in French.
Yeah, if you can, I would be happy.
My mother used to ask me, you should learn French. I never had the opportunity. Any question?
I think I have one. I can try to ask in English. It's good to practice. And first of all, thank you, Stéphaine, for your presentation. It was really interesting. And at the first part of the presentation, I got really interested about this molecule that you find out to have had reduced the tumor, which is peptide, right? And I was thinking about if this peptide could trigger some way an immunological response over the organism. Is that a possibility?
If this molecule could trigger what? Sorry, I didn't get the...
An immunological, I guess, I don't think it is the right...
immunological could be. The thing is, since the people I'm working with on this peptide, they are interested in the interplay between extracellular matrix and cancer. They've been doing tests related to cancer. So they do some what they call they look at migration. They do some scare and they look how it's close and repair. But so far, we don't have any experiment in our lab on the immunology part. But from what I could see as a presentation and the interplay they could be between these different effects, maybe if there were some tests, there would be a possibility to see something. But it's something we didn't test, so I cannot say yes or no for sure. But from my understanding on what people do, and I could see on conferences, we cannot say no for sure for that. We would need to test, yeah.
Right. Thank you.
Thank you, Mariana. I appreciate it. Any other question? Okay. You have an opportunity here because in this course you have students from different backgrounds, from different majors, okay? And yourself and myself, you came from physics and you work with biological systems. Could you use some time to tell people how it is to work in interdisciplinary research?
To work in?
Interdisciplinary, you know, when you make together two or more different research fields, physics, biology, computational, chemistry.
Yeah. In fact, it's very interesting that you say that because, in fact, the way we define our team, our group, so in our group, there's 9 PI, okay? And we have, now we have 3 PhD students and we have like 5 master degree students doing their training period. And we define ourselves as a transdisciplinary group. Because the thing is, I'm coming from physics. I have a colleague who's coming from physics. Most of our colleagues, they come from the biology and biochemistry field. Some of them, they turn to bioinformatics. And so we have one colleague that is from the cellular field, but is with people from other fields. For instance, Kevin, we developed the scoring function. He was doing his PhD between 2 different labs, our lab, so biology, and an informatics lab, and his salary was from a company. That's why in France we call that a CIFRE PhD students. The company paid for the PhD, so he would go from time to time to the company, and then he developed tools that are available for the community. And so we also have people from informatics in our group because there's a part that we developed that is dedicated to new representation of molecules in order to be able to communicate around our results, but also to represent chemical properties or structural properties on different surfaces and so on. So it's really something we try to keep and develop in our group. So of course, the difficulty at first is to find a common language and to be able to understand each other. But once if people are like willing, it can be long, but it's not so difficult. But then it opens up a lot of different ideas, and it's a very fruitful discussion. And in addition, we have collaborators from mathematics and informatics, and it brings a lot of things. So I'm really convinced that it's really the science we have to do if we want to find new solution and to be able to propose new research in the future. I don't know if that's answer your question.
Yeah, sure. Thank you very much. My idea with this question, because as I said, you have a lot of students for different majors here, and most of the time they are kind of afraid to start this kind of research, which involves physics, mathematics, computational, biology, chemistry, and say, I don't have all the tools necessary to carry out this kind of research, and nobody has. Nobody has.
No.
You have to share your knowledge and use this opportunity to learn. Yeah.
And sometimes it's even an asset not knowing everything about the field, because you won't have any barriers or you won't close yourself to solution. And sometimes it's because you don't know, you will say, okay, I will try this. And someone who knows the field would never try because he would say, oh, too much trouble, you will have this problem and this problem. Yeah. And sometimes it just opens fields. Of course, it takes time, but at least you've tried and you could find some solution or overcome some of the difficulties. So yeah.
And hopefully you are going to show this pathway to these young people, and hopefully you are going to have new students with me and with you.
Hey, that would be nice. That would be great.
Stéphaine, I really appreciate your participation. Thank you very much, and hopefully we are going to have a lot of paper, a lot of results together. Thank you very much.
Yeah, I hope so. Yeah, I strongly believe we will have. Yeah, for sure.
Thank you very much. I'm indebted
Thank you.
to you. Okay.
Okay, the same. Thank you very much for proposing. It was a nice pleasure. Great.
Yeah. Thank you very much. You are.
Thank you. So all the best, and we'll keep in touch. I'll tell you about the master degree students, and have a nice evening for all of you. Thank you for your question and interest.
Thank you very much.
Bye.
Bye bye.